to get going. It's my pleasure to introduce, we have actually a double introduction today because we have two speakers, and I'm going to introduce the first speaker, second speaker. First speaker will introduce the second speaker, something like that, yeah. But this is, we'll get to hear a bunch about frozen lakes today. It's a lovely time of year to think about it as the lakes are slowly thawing down here in Madison. And, you know, you maybe have wondered what actually happens underneath a frozen lake. Anything? And I think we'll learn today that there's probably more to it than you might think, especially if you're not somebody who's coming from the limnology world. So if you haven't met Professor Hilary Dugan, she's been here since 2018 at UW-Madison in the Center for Limnology, the Department of Integrative Biology. But Hilary's multi-talented, and her world intersects with many of us doing climate and ecology and land atmosphere sciences, and even projects related to just coding and development of ways to analyze large data sets, which is something that Hilary's an expert in. Prior to coming to UW, she had gotten her bachelor's and master's degree in Queen's University in geography and biology, and a PhD in earth and environmental science at the University of Illinois, Chicago. And since she's been at UW, she's been on a thread. I just pulled up her CV. She has 51 papers, pretty amazing. And just got an NSF Career Award on her work on lakes and salinity, so that's pretty exciting as well. So I'm going to turn it over. Hilary's going to introduce Ellie Socha, who is a graduate student who's working with Hilary, and so we'll hear from her.
This is great. I get applause, and I don't even have to give the presentation. Thank you. Yeah, so I set up as a double header, but over the last month, Ellie's taken over, which is great for me. But this presentation is going to be about a big NSF ecosystem project that we've had running for the last 3 years. Ellie's been a grad student on the project and has done a tremendous amount of field work, but also a tremendous amount of lab work, which you'll see today. So she's dedicated countless hours of her life to this. So I think it is appropriate that Ellie presents the results from what she's been doing rather than me. So I'm excited to have her do that. She's also going to be defending in a couple months. So this is sort of a nice presentation that culminates what's going to be in her thesis. And then in the fall, she's headed to Cornell for a PhD. So big congrats to Ellie for that. So yeah, I'm really excited to see this presentation, and hope you all too. And I'll hand it over to you, Ellie.
Good. All right, I'm gonna go micless here, but if you can't hear me on volume, just like give me 2 thumbs up, and I'll try to yell a little bit more. Thanks, Hilary and Anher. My name is Ellie Socha, and as Hilary said, I'm a second-year master's student in the Dugan Lab. Hilary and I work over at the Center for Limnology. I'm assuming most of us are familiar with campus, but if you don't know the Center for Limnology, just take a gander on down towards Lake Mendota, and if you run into a building with like 1960s architecture that looks like it's literally falling into Lake Mendota, you found the Center for Limnology. And apparently today is World Water Day, so I think this talk might be especially fitting because I feel like I've used the term limnology already a lot. So I'll be talking about limnology, which refers to the study of inland waters. And both the science and the term were coined by this person, Francois Alphonse Farel, in the late 1800s when he was doing some pioneering work on Lake Geneva over in Switzerland. Now, assuming once again most of us are familiar with Madison, I'm sure we all know how much our community loves and is interested in our water bodies, and that's rooted in our historical connection to the field of limnology. UW-Madison is often referred to as the birthplace for North American limnology, and this is largely due to these two people, Edward Burge and Chauncey Judet, who were doing some extensive field work on North Temperate lakes also in the late 1800s. They focused on the Yahara chain of lakes mostly, as well as a bunch of lakes up in the Northern Highlands Lake District in northern Wisconsin. Now, despite all of these people coming from temperate climates that experience seasonality, and despite all of them performing winter field work on lakes during periods of ice cover, the field of limnology as a whole has largely ignored the period of winter when forming our ecological theory and gathering data and understanding of how water bodies work on an annual cycle. Now, I'm going to ask you all to take a second and picture someone who's collecting data on a lake. Okay. I'm sure most of us pictured something like this. Here's a picture of me on Trout Bog, and you can see I'm in a boat. I've got my paddle and all of my equipment confined to the rowboat in the foreground here. I would have pictured something like this a couple years ago. Now I picture someone on an ice-covered lake. However, I think most of our associations with lake research have to do with this open water aspect, and we picture people on boats pulling water samples out from their boats. And this has historical roots as well, where open water or growing season research has proliferated. Winter research has really been left behind. And we see that this creates disparities in the amount of data we have on the winter season, as well as the literature and research that results from our data collection. Now, one figure I really like that illustrates these disparities is this query into the water quality portal data on algal or chlorophyll A observations. You can see there's a big difference between the gray squares or rectangles and the blue rectangles, which signify the winter field season. When we think of the months of December, January, February, March, and sometimes April, that's about 4 months of the 12-month calendar year. And if, like these chlorophyll A observations, other variables we collect mimic a same trend in the amount of data we have available, that's roughly 30% of a lake's annual life cycle that we have huge disparities in the information we know and can analyze. Now, these disparities translate over to the literature. One great example of this is a query into the Web of Science. They looked at papers studying lake algae. Of the 14,000 papers, only about 4% of them even mention ice in the abstract. And a further subsample of those 4% of papers found that only one of those papers discussed winter research on ice-covered lakes. Now, this seems like a big alarming deal to me. I don't know about you. And if we consider that there are an estimated 117 million lakes globally, about half of them experience ice cover during their annual cycle. So this amounts to some 50 million lakes that experience ice cover on a yearly basis. And I would reckon that most of them have data accumulated that's similar to these patterns. And this, in my opinion, sets up a potentially, it sets up a scenario where we could have a lot of blind spots, and our understanding of ecosystem interactions and the relationship between seasons is really constrained, and we can't make clear assertions about how seasons are connected to one another because we see these disparities in data and research available. Now, there are a lot of reasons that winter's been overlooked, and most of them are pretty reasonable in my mind. The first and maybe the most obvious has to do with the conditions that the winter season often brings about. Our primary concern when we're doing winter field work has to do with our personal safety. And in the wintertime, temperatures can get dangerously low, and we always try to remain dry and above the ice. To help, to help mitigate or protect us against unfavorable conditions, we usually limit our sampling activity to hours of daylight. If it does get warmer during the winter, that usually happens during the hours of daylight. It just sets up a safer situation. We also wear these really snazzy survival suits that are as bulky as they look to wear. They serve as personal flotation devices if necessary, so they literally are lifesavers. And I think every type of sampling method you would ever want to do on a lake is a little more challenging when wearing one of these survival suits, and there are some people in the room that can definitely attest to that. But we love them a lot, and we also love TikTok, so my sampling partner and I don't know. Okay, well,
It's going.
I'll play it one more time.
This is my outfit for today, and it looks like it's for me. Okay, love you.
So if any of you were were deep into the TikTok trends about a year ago. That was pretty fitting. Another reason that winter's typically overlooked is it can be challenging just to get to your field site. Oftentimes during the winter, we're dependent on local weather conditions and road conditions. And if you're lucky enough to drive right up to your sampling location, you still have a bit of a trek onto the ice and where you're hopefully trying to pull your samples from. For us, we often trailered our ATV with a truck to the closest access point via road that we could make it. These videos are just fun to show you what a beast our ATV was, and to give you a sense of what it's like to be a winter limnologist. Other times, and most times, we'd have to drive through the woods. A lot of the times we were on old logging trails. And if you know the North Temperate Lakes at all, part of the LTER research program, we often had to skip across Sparkling Lake, which in a vehicle that has a max speed of about 15 miles an hour, that takes a long time, especially when it's 15 degrees out and the wind is hitting you square in the face. And then finally, on days that things didn't go according to plan, Adriana and I would have to shuttle our 100+ pounds of field equipment through the woods, oftentimes through knee-deep snow, on these sleds. And I'll say, in all of my years of field work, I've never been more physically and mentally exhausted than after days like those. But it's all worth it. This brings me to another reason winter has been historically understudied, and that's because it's pricey. The startup costs come with a huge sticker shock if you don't already have a winter research program in place. I was curious the other day and looking up prices of our Argo ATV, and the baseline price is upwards of $30,000. And Hilary's told me that as you upgrade with different features, the upgrade costs are progressively greater and greater. And our ATV is an 8-wheeled amphibious ATV, so as I said earlier, we'd never try to go through the ice or get stuck in the slush, but we'd be safe if that had happened. And you can see it's decked out with some awesome snow tracks, which are totally essential. And then everything else in this picture is also very expensive. The survival suits are very pricey, the ice tent as well as ice auger. So if you don't already have a winter research program in place, these initial costs can be a barrier to performing winter field work. You don't need an Argo ATV, but I will say, having used an Argo ATV, it makes a world of the difference when you're collecting data in the field. Now finally, and probably the most reasonable reason that we've overlooked winter is because for a long time there was this assumption that as lakes cooled down and ice began to cover, processes in the lakes slowed down and reached a point of almost a stop. These scientists thought winter was a period of reset and thus inconsequential to open water and growing season processes. This figure is from the Plankton Ecology Group model, which was a pretty important paper back in 1986. And you can see over on the left-hand side of all of these squares that represents the beginning of the calendar year. And they're all starting from generally the same point of almost zero, where in this research, the importance of overwintering organisms wasn't appreciated, or well known, I should say, in springtime phenology and all of the events that happen in spring and summer. But it turns out in the last 15 to 20 years, there's been a lot of emerging research that tells us that winter is important to the life cycle of water bodies, especially lakes. Some examples of the importance of winter include this global synthesis paper, Ecology Under Lake Ice by Stephanie Hampton. And I think it's probably one of the most cited papers these days in winter limno research, even though it only came out in 2017. Here they found that on average, wintertime chlorophyll A concentrations globally were about 43% of summertime chlorophyll A concentrations. Similarly, phytoplankton biovolume, winter biovolume amounts were roughly 16% of summer biovolume. And finally, crustacean zooplankton densities reached up to 26% of summertime winter crustacean zooplankton densities. We also know that the timing of snowmelt in the surrounding area, as well as on top of a frozen lake, can correlate to summer chlorophyll A concentrations. If snow melts earlier, there's been studies linking lower chlorophyll A concentrations to north temperate lakes. For zooplankton, some groups even reach their maximum annual abundances when they're under ice, which kind of refutes the idea of that plankton ecology group model that everything comes to a stop, right? Now, this newfound interest in winter limnology research has only been heightened by climate change's predicted impacts to winter dynamics. One thing we really care about is snow cover, and we know that winter is changing and snow is changing. If we look at snow cover percentages, this study covers the late 80s to the early 2000s, and we see that in our neck of the woods, snow cover percentage had declined by about 15%. And if you need help orienting yourself, it's like we're looking down at the North Pole here. Similarly, full snow season is declining globally as well. The areas in purple and red are mountainous regions, I want to point that out. But in general, we see a lot of locations, especially like Wisconsin, that are seeing decreases in our snow cover season by up to 25 days. Another important variable that we care about is ice dynamics. This John Magnuson et al. 2000 paper is again one of the most cited papers in the field of winter limnology, where they looked at the timing of ice onset and ice breakup, spanning over 2 centuries of data, and found that ice onset was happening later in the season and ice breakup was happening earlier and earlier. Now, if we update that information with about 20 more years of data than the Magnuson paper had, we see that the changes to ice duration are even more pronounced and extreme than we once thought. Effectively, ice duration is declining at a rate of about 19 days per century for lakes that experience ice. And that has implications for lakes that experience intermittent ice cover versus annual ice cover. The orange dots on this plot represent water bodies that currently experience intermittent ice cover, and then as you move towards the North Pole, we see projected water bodies that will shift from having annual ice cover to having intermittent winter ice cover based on different climatic warming scenarios. All of Wisconsin is covered under 4.5 degrees Celsius increase and below. Now, if we zoom in on how Wisconsin's winter climate has been changing, we see over the last 70 years or so, our daily wintertime mean and minimum temperatures have increased by up to about 8 degrees Fahrenheit. In general, it looks like the winter temperatures have been increasing more in the northern part of our state. And when we look at projected changes in the next 35 to 40 years, it looks like that warming trend will continue in the winter, and more so in the northern part of the state. Now, if you combine the historical lack of focus on winter limnology, our newfound realization that winter is an important part of the life cycle for lakes, and that winter is changing, a lot of questions are emerging in the field. First, we're just trying to figure out what is happening under lake ice or during the winter in general, since we don't know the basics of a lot of these processes under ice. And secondly, we're trying to figure out how these processes might change in response to changing climate. If winter limnology interests you at all, I really recommend all of you go read this Cavalier et al. Paper. It's got a lot of good stuff in it and covers basically everything to some extent. However, the focus of my research and my thesis is mostly on the biological response to changing winter dynamics. Before we get into the specifics of our project, I want to make sure we're on the same page regarding those biological communities. So I focus largely on plankton under ice. And plankton are those free-floating organisms in water bodies. They're globally ubiquitous and abundant and really, really important. We've got phytoplankton, which are our plant plankton, off on the right-hand side, you can see some examples. And they form the basis of most of our aquatic food webs in terms of energy. They fix carbon and pull it into the food web in a useful form of energy. I think marine phytoplankton alone are responsible for fixing up to 40% of the carbon that's fixed from our atmosphere every year. So they're, I think, the textbook definition of tiny but mighty. And if you don't care about them yet, you should. We also have zooplankton. And zooplankton are the animal form of plankton. There's a bunch of different types, but for limnology research, we usually focus on 3 main groups. The first are cladocerans. An example of Daphnia, the genus Daphnia, is in the uppermost picture there. Daphnia are often considered keystone species, and they're model organisms in the fields of developmental biology and genetics. They range in size, but are usually pretty big. In terms of zooplankton, they can range from 1 to 5 millimeters. And a lot of focus is often put on cladocerans and Daphnia particularly. We've also got copepods, which are the ones with the black background. They're another large zooplankton. They've got really long life cycles where they go through a series of molting larval stages and several adult stages. As they get older, they're pretty effective predators and can be carnivorous. And then finally, our final group is rotifers, and these are much, much smaller. Think 0.1 millimeters is a sizable rotifer. And they, because they're small, largely because they're small, they have faster life cycles and reproduce quicker under ice. And they vary widely in the adaptations that different species have. So you can find a rotifer almost anywhere in any type of environmental conditions. Overall, plankton are really essential to food webs. If we look at a classic pelagic food web, we see that phytoplankton and zooplankton make up 2 key pieces of it. As I said earlier, phytoplankton form the basis of our food web, and zooplankton are a key intermediate in energy transfer, especially as you look to higher trophic levels like fish and birds and people. And if we go back to that historical assumption that winter was pretty inconsequential and all of this slowed down under ice, it seems really, really unlikely that that would be true. And my grandpa's been ice fishing his whole life. We know that under ice processes can support biology. So the idea that all of this would slow down and reset doesn't seem likely. And we know that these groups are active under ice. Phytoplankton can sometimes reach, they can bloom under ice. We know cyanobacterial blooms and phytoplankton blooms occur under ice pretty frequently. The zooplankton have a variety of ways to actively overwinter. It depends on their taxonomy and life history traits, but all of the 3 groups I mentioned have been found to commonly overwinter actively. So, I'm going to be looking at plankton under ice and how they respond to changing winter dynamics, right? And the idea in this project is a whole lake snow manipulation, where for two winter seasons, we plowed off the entire surface of this bog lake, which I'll talk more about later. Anytime we received an inch of winter precipitation, Paul Schramm and Noah Loddig and friends would get out there with snow plows and our Argo ATV that was attached to a plow and spend the day removing all the snow. This is clearly no small feat, and I don't think we can thank Paul and Noah and anyone else who helped out with that enough. And one thing I really love to watch in this video is track the sun in the sky. You can see that this took almost half of the daylight hours on a warm, sunny winter day. So, okay, we can move on. This is the general schematic of our hypotheses and predictions. From doing a snow manipulation and removing the snow, we're working under the general hypothesis that light is a primary driver of under ice processes. First, we think that removing the snow from the top of this frozen lake will impact ice dynamics. When we think about ice dynamics, there's black ice and white ice. White ice is formed from the wintertime precipitation, so things like snow and sleet, and it's generally really opaque and reflects a lot of light. Black ice, in contrary, is formed from the ice in the water column and is generally really transparent and can let a lot of light through into the water column. Thus, we think by removing the snow, that's going to decrease the amount of white ice that can be formed and increase the amount of black ice, thus increasing the amount of light that is transmitted into our water column. If we see more light transmission, we predict that there would be an increase in temperature due to more solar radiation reaching the water column. And we also think more light would support more primary production by photoautotropes that are trying to photosynthesize. So a lot of phytoplankton species. If we see more primary production, we believe that that forms a basis of more food and energy for our primary consumers, and thus we predict to have larger consumer densities. And when I say consumer densities, I mean zooplankton in this context. And then finally, more primary production might lead to more algal biomass and particulate matter in the water column, which generally decreases clarity. Secchi is a way that we measure clarity in the water column, and we think if there's more primary production, water clarity will decrease. To quickly summarize what we actually did, the 2018-2019 year served as our reference year where we didn't touch the top of South Sparkling Bog, we just took regular measurements, and then the next 2 winters served as manipulation years where every time we received that wintertime accumulation, it was plowed. So to start talking about our study site, we sampled South Sparkling Bog, which is in the Northern Highlands Lakes District up in Vilas County. It's about 3.5 hours straight north of here. On this map, you can see it mapped alongside the 7 North Temperate Lakes Long-Term Ecological Research Program lakes. I'm not going to say much about those aside from the rich research and data we have available dating back to the 1980s for the lakes that are in blue. I just want to point out the proximity of South Sparkling Bog to Trout Bog. They're really similar systems, and I'll mention Trout Bog later. But the distance from here to here is about 2 miles. So they're very close. Can everyone see that? Okay. Now the surrounding land use of South Sparkling Bog is essentially nothing. There's no shoreline development at all. There's maybe a small 2-lane highway that doesn't receive a lot of traffic, like a mile east, but other than that, it's pretty well forested. And though it's called South Sparkling Bog, it's not a bog, it's a bog lake, and that's because it's surrounded by this bog mat that's made up of sphagnum moss. And it's a dystrophic system, meaning that it's characterized by a lot of humic substances that refer to the amount of carbon in the water body. That can come from autochthonous sources, things like decaying organic matter within the water, as well as allochthonous carbon sources, things like decaying leaves that are eventually filtering their way into the water column. And that makes for a pretty acidic environment, and the water is generally really, really brown. This is humic water. Because of these characteristics, there's no fish. Environments like this don't tend to support a lot of biodiversity, so there might be a mud minnow here or there, but we're not going to talk about mud minnows today. It's dimictic, meaning that it mixes twice, once in the spring and once in the fall. And then finally, it's relatively small in area, 0.44 hectares. That's largely why we chose this system. If you don't want to plow off a super, super large lake, it's got a max depth of 8 meters and these other parameters, as you can see. And to give you a sense of what this looks like when you're sampling South Sparkling Bog, here's a video from our reference here. I want you to take note of the color of the water and how dark it is under the water. Here we're looking at white ice, so that ice formed from the snow and precipitation, and here's the boundary between white ice and black ice. It's pretty prominent in this video, but this is really, really dark. And you'll notice that even more when we contrast it to this video from the first year of our manipulation. Granted, this was taken on a sunny day, but you'll see much different ice dynamics here. Right now we're looking at white ice, and now we've reached the black ice, and it's very, very clear how clear it is and how much more light is entering the water. And I just love looking at all the little gas bubbles that have been trapped in the ice. It's a very cool video. props to Hilary for sending it my way. Okay, let's advance. So I'm gonna, if I can advance slides. Yes. Quick talk about sample collection, but mostly this is an opportunity for me to share really fun field pictures with you and convince you all to be winter limnologists. I want you all to appreciate how challenging it can be to drill holes in the ice that fit your sampling equipment. I feel like I got a lot stronger over my field season. And we can pitch tents and set up all of our equipment. And when we're actually ready, there's a number of variables and samples that we take. We did deploy an under ice buoy that collected measurements on temperature, dissolved oxygen, and light intensity roughly every 10 minutes at different depths within the water column. Manually, we collected snow and ice thickness measurements using a meter stick. You can kind of see it here. We also collected temperature, dissolved oxygen, pH, and conductivity manually. And this is a POV of what it's like to use a YSI probe on a frozen lake. We also filtered water for chlorophyll samples that were later processed in the lab, as well as we drew up water samples to do a bunch of water chemistry, not limited to these variables listed here. Some sampling dates we also measured nutrients as well. And then finally we sampled plankton. For phytoplankton, we just lowered a tube into the water column and drew up water, stored them in these dark brown amber bottles that were preserved in an iodine solution to inhibit any photosynthesis after we collected them. And then zooplankton sampling. This is my wheelhouse, so I love talking about taking a zooplankton sample. We use this thing called a Wisconsin net. Most people are surprised that it's called a Wisconsin net, and most people don't believe me. I haven't actually fact-checked that, but here at UW-Madison we call that a Wisconsin net. The main part of this I want you to note is the size of the mesh is 56 microns, so in theory anything larger than 56 micrometers should get trapped in the net and be included in our sample. To take a sample, we just lower it down in an upright position in the water column to our max depth for self-sparkling bog that's 7 meters deep. And the idea is to pull it up and capture all of these organisms and get a representative picture of all of the zooplankton in the water column. And we hope that they're all trapped in our net. This is what a zooplankton net looks like before you lower it into self-sparkling bog. One of the challenges of sampling zooplankton in the winter is if it's cold enough, your net freezes. So there were many times I'd have to go into our tent and actually hug the net to warm up the water so I could get all of the organisms concentrated down to the bottom part of this piece of equipment. This is what I'm looking at on the inside of the zooplankton. We call it a dolphin bucket. I don't know why. I like this video because you can see a little bit of movement from an invertebrate called Chaobarus. And essentially we transfer what we capture in here over into a glass jar and preserve it in 90% ethanol and later process that in the lab. These are just more fun field pictures of Adriana Gorski and I out in the field. Once again, you should all look into winter limnology. We had plenty of opportunities to look at really cool ice, and every sampling date was not complete without fun time to take a little lakeside nap or scoot around or mess around with the Argo ATV and enjoy some glassy ice. And this happened like literally every time we went out. Do some donuts. Alright. Laboratory processing. All of our water chemistry samples were worked up via NTL-LTR protocols. I'm not going to go into those more today, but if you're interested, I can send you a link on the specifics. Reminder, those are our variables. We sent our phytoplankton samples off to a company in Michigan called Phycotech, where they were able to classify the taxonomy of our samples and give us a bunch of different measurements, including abundance, biovolume, and biomass. Zooplankton were processed by yours truly on a Leica dissecting scope. This is the general setup that I had going over the summer. And sometimes what I'd see looking into the microscope for fun. My end goal was to identify all of the zooplankton to a species level. Disclaimer, that was very rarely possible in all cases. I was pretty pleased if I could identify to genus. And the general idea to do this is you take your zooplankton sample, you filter it through a small mesh, then put it into a beaker and dilute it to a known amount of volume. You take a subsample using this tool called the Henson stemple, and you put it on your counting tray and count, measure, identify, and then you can do some fun dimensional analysis to figure out what the relative abundance and density is by taxa. And Hilary told me I should include an estimate of how much time I spent doing scope work. I guessed about 250 hours, but the bulk of this came from me learning how to identify rotifers and getting comfortable with my samples so I could make a confident classification. I spent a lot of time looking at rotifers, and as a treat, sometimes I would find a cool rotifer and throw it onto a glass slide and put it on our Nikon compound microscope and image them at 400 times magnification, just because I think they're so pretty. And if you're going to appreciate rotifers, you have to see something like this. Take note of the scale. These are very, very small organisms, but I love this picture. Okay, we're going to get into findings finally. Before I talk about data we collected, just so you can get an idea of the winter climate during our study years, here's the air temperature taken from the Minocqua Airport. Minocqua is the nearest by city to South Sparkling Bog. We can see there's a little variability year to year, but in general they follow the same temperature trends. And moving on to snowfall, a little more variability, but the main takeaway from this figure is that every single year we did experience significant snowfall at some point throughout the season. Okay, now let's talk about what we found for snow and ice dynamics. So here's the breakdown. I'm not sure if you can see the white ice very well on this projection, but to me these look very different. Most notably, we see if there's any snow in our manipulation years, there's very little. We also see proportionally less white ice in our manipulation years compared to our reference year. However, I do want to note manipulation year 1, it does look like proportionally there's quite a bit of white ice here, but we attribute that to the timing of ice freeze. If you kind of zoom in here, our lake froze over in manipulation year 1 right around this time, and we received a lot of snowfall while it was freezing, so we effectively couldn't get out and plow the snow that was falling on top of the lake because ice conditions weren't thick enough and safe enough for us to do so. So that's what we're guessing caused this proportion of black and white ice. Now, if you don't believe me that the removing snow actually impacted our snow and ice dynamics, here's our South Sparkling Bog snow and ice plotted next to Trout Bog, that NTL LTR bog lake 2 miles away. That was sampled in tandem with South Sparkling Bog. And South Sparkling Bog is about half the area of Trout Bog, but other than that, they have the same max depth. Trout Bog is a dimictic, dystrophic bog lake, similar food web, all that good stuff. And the proportions of snow and ice look very different, right? And we see definitely that there is a lot more snow during our manipulation year 2 in Trout Bog. If you actually looked at an ice sample, you can see the differences between white and black ice. Here's the boundary between black and white ice from 2 different samples. I believe these pictures were taken on one of the early March dates, so maybe this date. But you can see the Trout Bog ice is a lot dirtier, cloudier, and has a lot more white ice than our South Sparkling Bog sample. We explore a little bit about temperature. This is temperature data from our buoy at the surface, which is technically about 70 centimeters under the surface. It's at that depth because we didn't want our buoy to freeze into the ice. It looks like our reference year was actually a little bit warmer earlier overall than our manipulation years. And one cool thing that we're going to explore in the future is the sudden increase in temperature prior to ice off in both of our manipulation years. I'm not going to talk more about that today, but here's the temperature data from the next depth down at a depth of 1.5 meters below ice. Moving on to light, immediately under ice, our light intensity is pretty substantial in our manipulation years, most notably in manipulation year 2. which is year 3 of our study. There are a couple of times when our reference year even shows up on this figure. I realize you can't see my mouse, but towards January and nearing May, you can see year 1, but overall, there's a lot more light reaching the water column in our manipulation years. And here's the light intensity at that next step of 1.5 meters. Moving on to our chlorophyll concentrations, we saw this general trend of increase in mean chlorophyll A concentration, both mean chlorophyll A and surface chlorophyll A, and trust us, the water was pretty green. If you don't know anything about chlorophyll A, it's often a proxy for plant growth and photosynthesis in water bodies. Our Secchi disk readings were, by and large, greatest in our reference year, as we predicted. This picture is not from our lakes. This is thankfully from USGS. Our lakes don't have that much green in them, but the Secchi disk is probably the most tried and true limnological piece of equipment. It's been around for hundreds of years and was created by some monk, I think, hundreds of years ago. So we've got a lot of Secchi data on a global scale, but our environmental variable correlation plot breaks down to something like this. And I've just circled a couple of the relationships we're interested in. Moving forward, I'm going to shift to talk a little bit about plankton and, in general, the environmental variables that we're most interested by are light, average snow, white ice, black ice, Secchi, and chlorophyll. So, first I'm going to talk about phytoplankton. This figure shows us the Shannon Diversity over time. And in community ecology, one way to describe biological communities is to look at its diversity. Shannon Diversity is one of the most common indices that we use, and it looks at both species richness, so that refers to the total number of species present in a sample, and it also looks at species evenness, so it takes into account the proportion of the total abundance that each of those species accounts for. We see that it looks like phytoplankton Shannon Diversity increased between our reference year and manipulation years. If we take a look at biovolume breakdown by taxonomic division, we can see a pretty steep increase in biovolume between our reference year and manipulation year. Most notably, there's a lot more chlorophytes, especially in year 3, or the second year of our manipulation. And the relative breakdown for total biovolume looks a little bit like this. What stands out most to me, first of all, is this cyanobacteria, or cyanophyta, which we're not going to go into that much today because cyanophytes are a beast of their own. But I'm really interested on this breakdown. We see a lot more chrysophytes and cyanophytes in our reference year, and they're distributed more evenly in terms of relative contribution to total biovolume. Now, one way in community ecology for us to explore variation in our community level data is to perform a canonical correspondence analysis. Hereafter, I'll call it a CCA. I'm going to flash this plot real quick. The point of this is to show you that if we perform a CCA on the lowest level of taxonomic classification that we have available to us, we get a bunch of points, and this is, in my opinion, pretty messy and hard to really extract much meaning from. And that's okay, because commonly in phycology, we work with morpho-functional groupings rather than purely taxonomy. And morpho-functional groupings give us more of a sense of the ecological niche that these communities are taking part in, or what they're doing for the processes under ice. Anyway, in total, there's about 11 groups based on Salzman Potissak, 2007. However, our data only showed 4 of them. And when you look at it this way, we see that there's a lot more mixotropes or potential mixotropes contributing to the overall biovolume in our reference year. Now, mixotropes are organisms that aren't purely phototropic. They aren't solely reliant on photosynthetic active radiation from the sun in order to photosynthesize. They can utilize heterotrophic processes and obtain their carbon from other sources, and they don't have to do photoautotrophy. Additionally, we see a big increase in the contribution from exclusive phototropes, which we might have predicted if we see more light transmission into the water column. There's more light availability for these types of phytoplankton to fix carbon and perform photosynthesis. Now we can look at the pretty canonical correspondence analysis. And to give you an overview of what this analysis actually does is it dissects our community level data and dissects it across all these axes and eventually tries to explain all of the variation with these axes that dissect it. We commonly see the results in this pie plot, and it shows us the first axis and the second axis. First axis always explains the largest percent of variation in our community level data, and then the second axis shows the next largest percentage of variation. And these axes take our environmental data and show us only what variation in our community data can be explained by the environmental predictor variables that we put into this model. If you're getting acquainted with the orientation and location of all of these, the arrows and the labeled boxes represent our environmental data, and generally we work in opposites. So as light intensity is increasing, we would expect white ice, average snow, SECI, and DOC based on this plot to decrease. Does that make sense? Okay. And then the points show us what types of groupings, so in this case our morpho-functional groupings, are most explained or related to those environmental variables. So to digest this plot a little bit, group 8, if you remember, were our exclusive phototropes. They're most related to increases in light intensity and black ice. On the other end, we see that our potential mixotropes, group 1 and 2, as well as this group of cyanophytes, were most related to increases in white snow, white ice, snow, SECI, and dissolved organic carbon at the surface. We're not going to dive into this group of cyanophyta because our range in temperatures were so small that we're not sure if there really is much there, so that's a question for another time. But we see that they're kind of in their own world up here. Okay, I'm going to quickly move on to the zooplankton community. The Shannon Diversity breakdown looks like this for our zooplankton community. It looks like it has a downward trend, and if anything, our zooplankton communities got less and less diverse between our reference year and manipulation years. Looking at the overall total individuals that I measured, the main point of this figure is so you can know that I was almost exclusively counting rotifers, so those really, really tiny ones. And if there was a cladoceran, it was literally one bosmina in a sample, in one of these two samples, pretty much, our first 2 years. So I was counting a lot of rotifers. And if we plot the taxonomic breakdown by genus, and we look at density, you see there is a huge increase in zooplankton density, but only really in the second year of our manipulation. I will note that all of these dates in year 1 and 2, there were zooplankton in all of the samples. However, the densities after the dimensional analysis came out to be between 0 and 1. So there were extremely small densities in these first 2 years. And I think first year of our manipulation, year 2, is the big question mark for me moving forward, why we didn't see such a huge increase in that year, but we did see this giant increase in density in year 3. If we want to look at the relative contribution to overall abundance from each genera, it looks a little like this. This is kind of a lot to digest again, and is a fun challenge in looking up the ecology of each and every one of these groups, which I'm still working through. My initial reaction to this is I notice Asplankna, which are the pink, and Ascomorpha, which are the orange. Asplankna seems to be pretty present in all of our samples, and then we have this huge increase in Ascomorpha in year 3. Once again, there's a lot of exploration and things that can be teased apart here. But when we plot this based on the lowest level of taxonomic classification that I was able to make, once again, we get a really messy and hard to digest canonical correspondence analysis. So another popular way we can group zooplankton is based on their trophy structure. And now trophy structure refers to its mouth and jaw parts, and it's really a proxy for feeding habits. In all, there's about 9 groups, or 9 different types of trophy structures. My samples only had 4. And here's the contribution for each trophy group based on density and relative average abundance. Nothing really sticks out to me initially when I look at this plot. However, when we plot a canonical correspondence of zooplankton community based on trophy structure, it comes out like this. Now, malleotrophies are doing their own thing up there. I'm not really sure what that has to do with, but malleotrophies are often categorized to be microphagous predators, where they create these little filtering currents with ciliated mouth parts that draws their food in, whereas vergate trophies and incudate trophies are classified as raptorial predators, so they kind of like pierce or grasp their food and bring it in. One thing that's very interesting to me is how vergate trophy 1 and vergate trophy 2 are straddling this plot in opposite directions. I think this is an area of exploration for me to look at the specific genera. The organisms that comprise most of vergate 1 trophy in my data were those ascomorpha, the orange ones on my previous plots. And they're known for being really effective at eating large algae. So there could be something there that explains why they're most related to chlorophyll and light. In the opposite direction, incudate trophy, in my data at least, most of these were from the Asplankna genera. And Asplankna are very, very large and omnivorous and pretty effective at utilizing cannibalism to obtain energy sources. So I think that could be another possible explanation or thing to explore on why they responded to those environmental variables the way they did. Okay, I'm finishing up, I promise. To get back to our predictions and what we saw happen, we predicted that removing snow would decrease white ice. We saw that. We saw an increase in black ice proportion and an increase in light transmission under the ice. We didn't see an increase in temperature between our reference and manipulation years. We're still teasing that apart, whether that's due to us removing snow, removed some insulation properties into the water column, or if just the climate and weather produced a colder or warmer environment under ice. We saw an increase in primary production through phytoplankton biovolume and greater consumer densities, especially in year 3, our second year of the manipulation. And we saw a decrease in density. I'm going to quickly restate what we just saw in all of my figures. Overall, phytoplankton had greater diversity in our manipulation years, as well as greater overall biovolume. And finally, we see some potential morpho-functional group shifts from more contribution from potential mixotropes that might dominate more in low light conditions to an increase in exclusive photoautotropes, so our chlorophytes, which are green algae. Our zooplankton community was made up of exclusively rotifers, essentially from the monogonata group. And we did see increases in abundance and density, especially in year 3. I think the big takeaway so far is that we saw more primary production under ice. Thinking about broader implications of these findings, we know that more primary production under ice could have impacts on what communities were promoted, especially as we scale up in trophic levels. This figure is from a paper that explored nutritional quality and the importance of fatty acid accumulation to successfully overwintering zooplankton. And the big takeaway from this paper was that algal fatty acids were the largest predictors of overwintering zooplankton success. Where they could acquire fatty acids from the microbial loop and terrestrial origin, it wasn't a big predictor in survival during the winter. Furthermore, that research has been expanded to show that the timing of Ison can be really important in the accumulation of nutritional fatty acids for overwintering zooplankton. Overall, the later that ice forms, creates a situation where zooplankton can acquire more nutritional fatty acids from phytoplankton right prior to ice formation, as well as in the early half of winter. And this has implications for springtime phenology. If we're supporting larger consumer densities over the winter, we have more grazing pressure right off the gate when ice melts. And that creates greater top-down control from zooplankton communities to graze on phytoplankton and algae in the springtime. If we revisit the plankton ecology group model, they updated this about 10 years ago, and you don't need to know the breakdown specifics of each color and line, but they basically incorporated overwintering populations and show us that based on overwintering populations and just trophic dynamics, the magnitude and timing of a lot of events are very variable, dependent on those characteristics. And finally, we think about increased primary production under ice. This is carbon moving out of the environment and into our food web. And if we scale up larger and larger, I don't think this necessarily has negligible effects to carbon cycling. I'm also not aware of how many carbon budgets really pay attention to winter. So that's food for thought. And then the study adds to the body of knowledge on plankton under ice. You can see there are not many studies out there that explore plankton under ice in lakes. And this one's unique in that we have a dystrophic study site, excuse me. That about wraps it up, but I have to give my acknowledgments to the Dugan lab, my advisor Hilary, Adriana Gorski, who's my lab mate and taught me most of what I know about actually being a winter limnologist in the field. Emily Whitaker was the grad student that got the ball running on this work, as well as Ella Schmidt and Sydney Wydell for help in the field. I see Liz Rundy here today, and I forgot to include her on our slide, but she helped process a lot of our water chemistry data, so thank you, Liz, and the LTER crew. My committee members, in addition to Hilary, are Jake Van Der Zanden and Gretchen Garish, and they've given me a lot of guidance over the last 2 years. It's funded by NSF and UW-2020 and Anna Grant Birch. And then finally, my parents, Tracy and Jim, who are the best people in my life. And that's it.
Thank you. Thank you for just a fantastic and fun talk with a lot of great pictures, diagrams, all sorts of food for thought. Questions from the audience? And I'd like to always start with students. Do we have any?
Yeah.
Is there a reason that you would expect the rotifers to have the largest response out of all the zooplankton? And would that, is there a fast response and then a slow response? Like, would other groups kind of catch up if you did this for like 10 years?
That's a really good question. I'm not sure about the aspect of other groups catching up because the under ice community structure, it seems like, is very dependent on fall conditions and early ice dynamics. But I would expect rotifers to respond more immediately because their generation times are so much smaller for a lot of organisms that they're maybe 4 to 5 days. So they can respond and adaptations can, or useful adaptations can become more prevalent much quicker. The copepods have really long life cycles of up to years and months. And cladocerans, while they actively overwinter, they're not generally associated to be a primary group relative to the other zooplankton groups for under ice seasons. Did that answer your question? Kind of?
I think so.
Okay.
Yeah, I mean, I don't know very much about this, so.
That's okay.
Thank you. Other questions?
Yeah, hello.
So one thing that stuck out to me was that almost that 1 year lag period from manipulation year 1 to manipulation year 2 in both the phyto and zooplankton. I know you said you're still working through it, but do you have any working theories on why there is that lag?
Kind of been going back and forth with that. I think an obvious answer that I don't yet know how to parse out is the nutritional quality of the phytoplankton that increased between year 1 and year 2. If you recall the phytoplankton biovolume figures between all of the years, our year 3 had a substantial greater amount of biovolume than even year 2. It probably had double. So maybe that's something to do with the abundance of food availability, but also the nutritional quality. I was kind of weirded out when I was working through the samples, and I worked backwards when I was processing these. So I started with 2021, then went to 2020, and I was like, wait, I thought these would be more similar, and they weren't. So that's definitely a big question mark, and I think an exciting area to hone in on when I'm actually writing this thesis.
Yeah, I was struck too by that the 2 manipulation years also had a pretty large difference in total light availability under ice. I don't know, how much of that was just chance, or or is there some sort of that going on based on the amount of snow that fell that year?
I think if you think back to the proportion of white and black ice between the year 1 and year 2 of manipulation, we did have a substantial greater amount of white ice during year 1 due to the timing of our ice formation. I think that had a greater influence on light availability between the 2 manipulation years.
It was all freeze up bunker.
Yeah.
It froze terribly the first year.
I
The
do remember that.
snowstorm.
Yeah.
And then this year was last year.
Let's take one last question.
Yeah, great talk. I have one comment and one question. I wouldn't be surprised that the temperature didn't go up, because for my expertise is in sea ice, which is quite different, but the way that the temperature is set at the bottom is a lot more by conduction of heat rather than the radiation that penetrates the ice. And if you have a snow cover, it will isolate that very nicely, and so it keeps it warmer underneath if you have a cold winter. Whereas if you don't have that snow cover, the ice is really good at conducting that heat down, so
Okay.
you'll grow. And that's also why you see much more ice growth, right? Because that black ice grows at the bottom, and so that would explain why
That
it
makes
goes
sense.
colder. And then related to the other question, sort of like, do you think you have like a preconditioning from your first year, of removing the snow, or do you think your system totally resets for the second year?
I'm not sure what my stance is on that. Winter to winter, there's such a long amount of time that separates the two. In plankton ecology, we know that seasonal communities are connected to one another, and we can see cross-seasonal relationships that are impacted, especially by warming climate. I think to the degree in that the first year's winter surviving group impacted the spring and summer group, which impacted the fall group. Those were connected, but I don't know to what degree we could really figure that out yet. But that's something I've been thinking about, or will continue to think about.
Yes, lots to think about. And be curious too what happens if our Yahara Lakes this winter, which had a very low amount of snow on it for much of the winter. I'd be curious to see how any of those holds up. Let's give another hand to both. I do want to just let you know about there will be no CPEP next Tuesday. But the Tuesday after that, April 5th, we have another speaker coming from out of town. Her name is, Liz, you got to help me with the last name, Alice du Vivier. Yep. From the National Center for Atmospheric Research. She'll be visiting, and we'll be meeting with folks during the day too, as well. And so I'll talk to Professor Maroon if you want to join up on any of his meetings. Her talk is going to be about sea ice. So change is rough, the impacts of changing sea ice on Arctic winds. So we'll continue our ice trend here as we move into April. So thanks again, and see you in 2 weeks.